This video goes along with Chapter 10 notes, which will cover photosynthesis. So our focus is to look at the energy changes that occur during photosynthesis. And this is the essential knowledge that by the end of this and going through all of your classwork, you should know, hopefully. Starting off with an overview of photosynthesis. Plants and other photosynthetic organisms contain organelles called chloroplasts. Some of the more basic organisms will just have they don't necessarily have chloroplasts, but they have photosynthetic regions, which gives us a clue about where chloroplasts came from. Autotrophs are organisms that can sustain themselves without eating anything derived from other organisms. Essentially means they can make their own food. Autotrophs can produce organic molecules from carbon dioxide and other inorganic molecules. Almost all plants are photoautotrophs, which means they're using sunlight to make those organic molecules. There are a couple of exceptions for that. Some things use sulfur instead. Photosynthesis occurs in plants, algae, other unicellular eukaryotes, and some prokaryotes. This is what the chloroplasts look like, which we'll get into a little more detail in a few minutes. And these are some examples of photosynthetic organisms. So, of course, we have plants, multicellular algae, unicellular eukaryotes, cyanobacteria, and purple sulfur bacteria. And all do photosynthesis in their own way. Overview of photosynthesis again. Heterotrophs obtain organic material from other organisms, which means that we depend on autotrophs for both food and oxygen. Photosynthesis captures energy from the sun and produces sugar, which means it is converting solar energy or light energy into chemical energy. The reaction for photosynthesis is 6CO2 plus 6H2O plus light energy yields C6H12O6 plus 6O2. That means that 6 molecules of carbon dioxide and 6 molecules of water with light energy can make 1 molecule of glucose and 6 molecules of oxygen. The goal of photosynthesis is to make glucose. Oxygen is a byproduct. It happens to be useful to us and subsequently for the plant for the purposes of respiration, but it is not the main purpose. Photosynthesis occurs in 2 steps. The first one is called the light reaction or light-dependent reaction, where water is split to release oxygen gas. And the second part is called the Calvin cycle or the light-independent reaction, where CO2 and high-energy electrons are used to make glucose. Let's backtrack to where did photosynthesis even come from. Photosynthesis first evolved in prokaryotic organisms. Scientific evidence supports the claim that prokaryotic photosynthesis was responsible for the production of an oxygenated atmosphere. So one of the things we know from looking at lots of different types of data is that the composition of our Earth's atmosphere has changed dramatically over the last 4+ billion years. At one point, several billion years ago, there was an oxygen revolution where our atmosphere started to contain more oxygen, obviously, and that's from prokaryotic photosynthesis. We also know that prokaryotic photosynthetic pathways are the foundation for eukaryotic photosynthesis, which leads us to the theory of endosymbiosis, which essentially says, and we can look at this when it comes to mitochondria as well, is that both chloroplasts and mitochondria a long time ago were their own individual things that could be alive on their own. And then some sort of pre-eukaryotic cell consumed that thing, and instead of it being destroyed, maybe the cell somehow figured out it was super useful to have a chloroplast or a mitochondria, and then that gave way to our eukaryotic pathways that we have now. We know mitochondria had to come first because all eukaryotic organisms have that, but not all have chloroplasts. Some other pieces of evidence for this are that both chloroplasts and mitochondria do have that double membrane, where other structures do not. They also contain their own ribosomes that are more like the size and shape of ribosomes found in prokaryotes. And they even have their own DNA, which is circular. And prokaryotic cells do have that small circular DNA. There's lots of evidence telling us that this is what has happened. Let's take a closer look into chloroplasts. Leaves are the major locations of photosynthesis in plants because they're going to have the best access to sunlight. Chloroplasts are mainly found in mesophyll cells, which are the interior leaf tissue. Like they're like dead on right in the center, and each one will have between 30 and 40 chloroplasts. Also on the leaf, CO2 enters and some O2 exits the leaf through stomata, which are like pores or openings on the underside of the leaf. So we have gas exchange from there. Light comes through the top of the leaf to strike the chloroplast, and then water can enter the leaf through the vein. So it's coming up from the roots to the vein, and then sugars will also leave the leaves. Sorry, that seems complicated to say. Leave the leaves through the vein as well, because we know we don't want to keep all of the sugar in the leaves because it is useful for lots of different cell functions and structures. So here is our chloroplast. Again, it has an inner and outer membrane. There's a fluid inside called the stroma. And then also inside we have thylakoids, which are connected stacks in the chloroplast. Each grouping or stack of thylakoids is called a grana. And there also is the pigment chlorophyll that's found on the thylakoid membranes. And the leaves actually appear green because chlorophyll reflects and transmits green light. There are 3 types of pigments that are found in chloroplasts. Chlorophyll A is a key light capturing pigment. The absorption spectrum of this pigment does suggest that violet, blue, and red light work best for photosynthesis, which you can see here from the graph. So you may notice that lots of lamps that are meant to help plants grow are in that color range. Chlorophyll B is an accessory pigment, and then we have carotenoids, which are a separate group of accessory pigments, and those help broaden the spectrum of color that drives photosynthesis, and they can absorb excess light that would damage chlorophyll or react with oxygen. And since it is fall, we notice that leaves are changing, so chlorophyll, our normal green pigment, is not working as much, and so what the leaves show in color is usually from the carotenoids that they have and whatever other accessory pigments. So we see their absorption because chlorophyll, the green one, is not just taking over and only showing that. That was a long aside, but it's fall, and anyway, there we are. So to sum up, this is a zoom in of our leaf, and then you can see the leaf structure on the bottom. Let me get my mouse out here. This would be the stomata where we're having gas exchange. And these cells right here are the mesophyll where more photosynthesis is taking place. So we have our cuticle and upper epidermis here, lights coming, striking down through here. And then this purpley thing right here is our main vein. So, we have a light coming in through here. This is where most of our photosynthesis is taking place, close to the vein. And then we have gas exchange on the underside of the leaf. And then we zoom in and, even more to chloroplast. And that's a for real image of a chloroplast, not a cartoon drawing like you're used to seeing, but that's a for real thing. Moving on to talk about the light reaction. As an overview, thylakoids transform light energy into chemical energy, which here is ATP and this thing called NADPH. Water is split during this reaction into O2 and hydrogen electrons, and those hydrogen electrons will be used later in the Calvin cycle to help make glucose. The hydrogen electrons are added to this thing called NADP+, which makes it NADPH, and all that is an electron acceptor. So having those high energy electrons floating around in the cell is not going to be something that anybody wants. And so we have those special electron acceptor molecules that can take on those electrons to be in a more stable form, and then they'll be taken off NADPH during the Calvin cycle. Also, during the light reaction, we're able to generate some ATP from ADP by phosphorylation. What comes next is going to be a lot of words, so buckle up. You're going to be okay. We're going to get through this together. All right. So we're going to go into a lot more detail about what happens because I'm assuming what was on the slide before this is probably where you stopped in Bio 1, and that's totally fine. That's where you should have been. When a pigment absorbs light, it goes from a ground state to an excited state, which is unstable. When the excited electrons fall back to the ground state, excess energy is released as heat. So we have this thing called a photosystem that consists of a reaction center complex and light harvesting complexes. The reaction center complex is an association of proteins holding a pair of chlorophyll A molecules and a primary electron acceptor. The light harvesting complex consists of pigment molecules that are bound to proteins, and they transfer the energy of photons to the chlorophyll A molecules in the reaction center complex. A primary electron acceptor in the reaction center can accept the excited electrons, which here would be NADP+. So here's a picture of that. We have our light harvesting complexes and then the reaction center complex in the center. That square thing is our primary electron acceptor. We have our pigment molecules, and you can see the transfer of energy as the electrons fall from their excited state. And that's all for that. There are two types of photosystems in the thylakoid membrane. There's photosystem 2 and photosystem 1. Photosystem 2 functions first. The only reason why it's not called photosystem 1 is because, so in the pathway, technically the second photosystem was found first, so it got called photosystem 1, but then they found, like, maybe just photosystem, but then they had found another one and just called it 2, but photosystem 2 is what happens first. Whew. The reaction center chlorophyll A of a photosystem 2 is called a P680 because it's best at absorbing a wavelength of 680 nanometers. And photosystem 1 is best at absorbing a wavelength of 700 nanometers, so its reaction center is called P700. During the light reaction, there are two possible routes for electron flow, linear and cyclic. Linear is the primary pathway, which involves both photosystems and produces ATP and NADPH using light energy. With a cyclic electron flow, electrons cycle back to photosystem 1 reaction center. So it's only using photosystem 1 and produces only ATP, so we have no oxygen being released. Let's go through linear electron flow first. So a photon hits a pigment in a light harvesting complex of photosystem 2, and its energy is passed among pigment molecules until it excites P680. An excited electron from P680 is transferred to the primary electron acceptor, which is called P680+. Water is split by enzymes, and the electrons are transferred from the hydrogen atoms to P680+, reducing it to P680. And then each electron falls down an electron transport chain from the primary electron acceptor of PS2 to PS1. Energy is released by the fall, drives the creation of a proton gradient across the thylakoid membrane. Potential energy is stored in the proton gradient, drives the production of ATP by chemiosmosis. In PS1, or photosystem 1, transferred light energy excites P700, which loses an electron to the primary electron acceptor. And then again, we have each electron falls down an electron transport chain from the primary electron acceptor of photosystem 1 to the protein FD. And then electrons are transferred to NADP+ to form NADPH. I hope you're okay. You may want to read through that a couple times and check back in with yourself because that's a lot. So here's a picture of that. So 1, we have light striking, electrons moving. We're going down the electron transport chain twice, and then finally we get our final product of NADPH. Here's that at a larger view, where you can see the whole process of the light reaction in all of its detailed glory. Moving on to the Calvin cycle. In the Calvin cycle, plants use carbon dioxide, NADPH, and ATP to make sugar. It is an anabolic process because we're building sugar from smaller molecules using ATP. It regenerates its starting material after molecules enter and leave the cycle, and this process happens in the stroma of the chloroplast, and that's that fluid that's on the inside of the inner membrane. Carbon enters a cycle as CO2 and leaves as a sugar called glyceraldehyde 3-phosphate, or G3P. For net synthesis of one G3P, the cycle must take place 3 times, so it's fixing 3 molecules of CO2. Later on, so anyway, we have to make 2 molecules of G3P, and later on those will be used to make glucose. The Calvin cycle has 3 phases: carbon fixation, which is catalyzed by this thing called rubisco, reduction, and then regeneration of the CO2 acceptor RUBP. So here we have an input, 3 CO2 entering per 1 cycle, and this is going to have to happen twice to make 1 molecule of glucose. So we have carbon fixation happening first, so we're changing all of this stuff, and then it gets reduced to G3P, which exits the Calvin cycle to form glucose and other organic compounds, and then it's going to regenerate the stuff that it's used so this cycle can happen over and over again. The takeaways from this, am I expecting you to have every single little detail memorized? No, but I do need you to understand the main phases and generally what's happening here. So we have to fix carbon, and then it gets reduced to form GP3, which exit the cycle, and then we regenerate everything that we've used. So maybe a little more detail than that, but don't let this hurt your brain. Let's review what we've learned about photosynthesis thus far. The energy entering chloroplast as sunlight gets stored as chemical energy in organic compounds. Sugar made in the chloroplast supplies chemical energy and carbon skeletons to synthesize the organic molecules of cells. Plants store excess sugar as starch in chloroplasts and other structures such as roots, tubers, seeds, and fruits. The storage piece is important. We're not immediately using all of this glucose for respiration. It does have to be made for other structures and processes as well. That's a review of our photosystems. Maybe, this diagram is more helpful for you than the other one. This is I think, better view of the Calvin cycle. It's a little cleaner without all of those intermediates that are used. And then a comparison here of the light reaction in the Calvin cycle. So the light reaction again happens in the thylakoid. We're converting light energy into the chemical energy of ATP and NADPH. We're going to split water and release O2 to the atmosphere. Maybe plants will keep some for respiration. The Calvin cycle, on the other hand, takes place in the stroma, and they're going to use ATP and NADPH to convert CO2 to the sugar G3P, and then ADP plus the phosphate and NADP plus will be returned to the light reactions to pick up, more electrons and turn back into ATP, which I think, honestly, one of the most incredible things to me about this process, and later when we talk about respiration, is how good organisms are at recycling things that they need to use over and over and over again. So we're not having to get a new NADP plus every time photosynthesis happens. We're using, plants are using what's already there. So that is photosynthesis in a nutshell. And to sum it up, let's put all this in a chloroplast. So we have a light and water coming into the light reaction with O2 coming out. It's also going to make ATP and NADPH, which gets sent over to the Calvin cycle. CO2 comes into the Calvin cycle. G3P comes out, and then NADP plus and ADP plus P get sent back to the light reaction. And I know, looking at this, the details aren't there, so maybe this is easier to look at, but know this and also the details.